Welcome to this session about our EU project WaSeaBi. We are several people here working in the WaSeaBi project. We have Mehdi, Carlos, Jone, and also Paula sitting here, and myself, Grethe, who will present this EU project. We are really a lot of people working in this project, so this is only us who are here to present the project today and how far we are. So I will begin by telling you the overall of the project. So it's called WaSeaBi, and what we are dealing with here is the side streams from the seafood production. And we want to help the industry to get more out of what is sometimes looked at as waste, but is more side streams and can be used for very many different things. And we are looking into that in this project to both how it is in the industry while handling the side streams, but also some of the processes and technologies to valorize these side streams. So this is an EU project, as I told you, and we began the project in 2019 and will end it next year in September. And you can see here how many, or the budget for the project. And to show you how many people or how many different groups there is a part of the project, you can see there is both people from the north, the middle, and the south of Europe. And it is both the universities, but also industry, which are taking part of this project. The main objective is to solve the challenge that prevent sound exploration of the aquatic resources. So we want to be able to get much more out of what we are actually having in the seafood production. So both on in the industry by looking into the sorting, but also by the storage technology and also the technology to take out the proteins and the lipids and so on and use it in other types of either food items or into feed or biogas, of course. That is also an issue. So everything is used when you are dealing with the seafood or the raw material for the seafood. Here we show you what type of side streams that we have in the project. Of course, in such a project, we cannot take in all side streams there is in the seafood industry, but we have tried to span so that we have different types of side streams. You can see that we have both the mussels, and there we are working with both the boiling water, but also the shells. We are working with different types of, or different species of fish, which is important in the different areas of Europe, but also with the brine from the salted cod. And we have looked into all the parts in these production lines. It is Professor Charlotte Jacobsen who is the coordinator of the project, and you can go to the website to find much more information about the project. And of course you can contact the people working there, either one of us or Charlotte or one of the other persons, researchers or industries that are in the consortium. So actually today we also want to involve you. So we have made a Kahoot, and I will try if I can get it to work now. This is, let me see if I cannot see it here on this screen. So, you are going to need your mobile phone. Der var den. So Probably here. I think I need help.
I don't know if I will be able to.
I cannot find the mouse.
But it's there.
Is
So
it there?
can-
But I cannot see the. Oh, okay. I hear.
Try to
So
go-
yes,
yeah.
there. Okay.
That's the only
Good.
way.
Yes. Fine.
It's
We
working.
manage. Yes. So. Yeah. Okay. Now there is this QR code, so you can actually scan it with your smartphone, or you can go to the website. So scan it, and then it will ask you to type in a name, and we will be able to see you here in a short while. See, we have already one person. Yes, one more. One. Yeah. There's room for more. Yeah.
Oh, they don't have aquaculture.
Yes.
If they cannot scan, they can go to the home page and add the code.
Yes, you can go to the code page and add the code.
Just Google Kahoot, and then you will end up to there. You add this pin code, and you will go to this page.
Yes. Okay. Should we try to continue and then?
Some people are trying. Some people are trying.
Okay.
QR code system.
A little more. Yes. Oh yeah, you can see it here.
Yeah, you can also click on the menu. You don't have to tell me anything.
Yeah.
It's not the same QR.
No. It's not the same QR code as on the postcard.
No, you have to use that.
I will tell you about the postcard later.
Yeah.
Yeah.
Okay.
Yeah.
That's why.
Adam, can you use?
Oh,
This is for you.
sorry. Yes. Yes, for the newcomers, we are going to make a Kahoot. So if you want to join us, please scan the QR code. Yes. The one on the screen and not on the postcard. Mm. We will start the Kahoot now. So be ready for the first question. Yes, right now you type your answer. Yeah. Yes, I will try to, there's more. Okay. And we can see here. So there's people from both Canada and Norway, and Spain is of course here. And the Netherlands, Greece, and Ireland. Yeah, so that's nice to see people from all over the world. So we go to the next one. So this is about the species, and please write it in English, the name in English. Yeah. Good. Yeah, salmon is one of the big products here, but also mussel and shrimp and turbot. So this is good to see that also some of the species that we are going to, or we have been working with, is a part of this. We will stop it now and then have a little more presentation, and we will come back to this. So I will need to go back here. And how do I do that? No mouse.
I don't know what to do. What is that?
It's, yeah, it's,
I have to.
Tranqui, tranqui.
Pero esto no hay que cerrarlo, hay que minimizarlo. Luego lo vamos a seguir usando.
Vale, pero hay que ponerlo en pantalla completa aquí. Espérate, se nos ha ido aquí. Vale, ahora lo tenemos ahí. Y ahora lo único que hay que hacer es abrir aquí.
Eso es lo que estoy
yes.
intentando.
But the mouse was not there, so yeah.
El ratón se va a dañar.
Okay. One of the questions that we, of course, ask is, would the seafood industry like to turn their side streams from their seafood production to into higher value ingredients? So, and when we have talked to the seafood industry, this is also what they say that they want to. Of course, that will be sustainable, and it will also make a good business to valorize the side streams. So therefore, we need information about how the side streams are handled now, and also, and also what can be done, and therefore we have this questionnaire. To know what is done already now, and I will come back to this questionnaire in a short while. We have these work packages that you can see beneath on the lower part of the screen, and what we want to do in the first work package was to look into what is done right now, which are the side streams that exist in the seafood industry, and what is done with it, and also to look into the consumers' attitudes towards using these side streams in food products. But that's another story. Today we are talking about the industry. Besides asking about what's already there and how it's handled, we also made a characterization of the side streams. And what we got from the first questionnaire we had, it was a very long and detailed questionnaire. We got the answers that there's a lack of technology to handle the side streams, and also space and personnel for handling these side streams. And also this by sorting and storing is a part of the project that you will hear more about. Also lack of market and final products. And that was why we also went out and asked the consumers about their attitudes towards this. But as I said also before, we looked into what is the characterization of the side stream, and also to get an idea of what is the variation in these side streams. So you can see here all the analysis that we did of the side streams that we collected, and you will hear much more about the results from all these, and also results and what have been done with the technology to handle these side streams. But we really want to know more about the side streams and how it's handled out in the seafood industry. And therefore we have made a shorter version of the very detailed questionnaire. And here, this is a QR code that is on the postcard that was on your seat. And please help us help you by answering this questionnaire. You don't need to do it now. You can do it whenever you want. So, and these results will only be used in this research project. And so we would really want to help you doing this. And by this, I would give the word to Mehdi to continue.
Thank you, Grethe. Yes, the first question that comes up, if you want to use these side streams as a resource for production of different products, is that are they already handled in a proper manner or not? That we want to use them. And here I will talk a little bit about the sorting, which is the primary step for utilization of these resources, and the stabilization technologies, which would be the second requirements. If we look at the status already in the seafood industry, this is the case that we have had for herring at least. I know that in some industries like salmon and cod, the situation is a bit better. But for herring, they are normally combined together in a bin, like what you see in this figure. And of course, it would be a mixture of everything: blood, fat, enzyme, natural flora of microbia. And imagine when these come together, what will happen? It would result in, of course, cross-contamination, lipid oxidation, and microbial growth. Then you will immediately lose the large part of the value of your side streams, which are sometimes very nice start materials. In the WaSeaBi, we have tried to, of course, work on this, and we have worked on a filleting machine. We have redeveloped it, and we have managed to fractionate and separate these side streams, which allows to get the belly flap separated, head in a separate part, backbone, and the tails. And you see the pictures, how clean and nice they will be compared to the previous picture. And now instead of one product, which was the fillet, you will have 4 or 5 fractions that are ready for value addition and for the next step, which would be either protein, fat, or different compounds to be extracted. Then the first step has been successful, But the next step is that these materials are very sensitive. They are full of omega-3, which are sensitive fats. Still, there are a lot of blood in this, which can easily, by exposing to high temperature, act like a bomb and get oxidized or rancid or spoiled. To work on this challenge, we have developed a dipping technology that can be reused for up to 10 times. You can get the side streams that you can see in the picture. They can be dipped into an antioxidant-rich solution, and then they will become much, much more stable. The rest shows the experimental design that we have had. What it would mean in terms of stabilization, if you don't dip the side stream, they will rancid in 1 day. They will not be useful anymore in case of herring. But if you dip them in different solutions that we have developed, you can see they can be stable up to 10 times longer compared to the classic storage that is already common, let's say, which is a very big achievement. As I mentioned, this antioxidant-rich solution can be used up to 10 times, which will reduce the cost substantially compared to a single-use system. Then we have tried to use a little bit frame fraction. It is already implemented in the industry. They get the frame. They have already made the line to do the dipping even in the industry. They are dipped in the antioxidant solutions, then they are taken into a mechanical separator. You don't see my mouse. I don't have it here, but it's okay. Then they come to the mechanical separator, and then they would be a very nice mince, which is more like a meat coming from the ocean, and it's ready for production of different products. It has been tested already for production of fish burgers, which have been served in different events. And now another company is trying to make these burgers and sell them inside Sweden. And of course, the mince that would be sold out in different countries. Then, of course, there has been a team involved in this, and you are welcome to contact us if you have more questions. I must invite Grethe to continue the Kahoot, right? Before we go with Kahoot.
So,
Yeah. Then I will invite
go to the next
Kahoot.
speaker.
Okay. Thank you. I will continue this talk speaking about some of the technologies that we have used to produce other kind of products, which applications and potential market we will speak later. My colleague Jone will speak later about it. Basically, we have worked, as you have seen in the previous presentation, in whole fish, in fish discarded, in our case was hake, under the minimum conservation reference size. Also, the people in DTO have worked in herring, and we have worked in liquid side streams from the cooking process of mussels, and also in other solid processing side streams like from salmon and also from cod, you have seen in the previous presentation. What kind of technologies we have employed? We have now at pilot, we have worked at lab scale, and now we are planning upscaling at pilot level to produce several products. One of the technologies employed, you probably know, it is the filtration through membrane technology. More specifically, the Nano Filtration that is represented in the figure. Basically, you have the product, this liquid solid side stream that is filtered. you retain the molecules that are interesting, and the permeate, what crosses the membrane, is the water that you can reuse and recycle. You can use this membrane technology to concentrate, as you can see in the figure. Just to concentrate, you eliminate the liquid part, and you retain the molecules that are concentrated in the concentrate. And the permeate is this water that you can reuse. If you have salt, that is the case with many of these liquid-sized streams, you can use the same technology in a diafiltration mode. What means that you concentrate, and after that you can dilute several times the retentate to dilute the salt, and in several cycles of diafiltration you maintain the concentration of the part of that interests you to recover, the molecules that you want to recover, proteins or lipids and so on, and you dilute and several times the salt, and you always obtain this water that is clear and can be used, reused. This view represents the products that you obtain. You have the original sample in the center of the screen. You have the concentrate or the diafiltrate, that is always this fraction rich in molecules that are interesting. And you have this permeate that crosses the membrane, that is basically water with salts and very, very small molecules. The other technology that we have used with the solid byproducts is the hydrolysis. Protein hydrolysis can be done through these technologies. There's the silage, that probably you know it. Technology that is as old as you can see in the figure. The Romans used it already to fermentate the fish together with their viscera, where you have enzymes that digest the fish and produce a ferment that was very appreciated by the Romans as a flavor compound. The nowadays version, I do not want to do publicity, is that you can see it in this bottle without publicity. That is also a fermented product that uses whole fish and the enzymes of the fish to produce flavor. And the result of the hydrolysis of the protein, that is represented as a chain of amino acids, depending on the intensity of the hydrolysis, using silage or using, as you can see here, using commercial enzymes, depending on the intensity of the hydrolysis, you can obtain peptides that are pieces of protein that can have bioactivity, that can have some health activities, and that can be used in products like nutraceuticals. Or you can also make a more intensive hydrolysis, producing more smaller pieces of peptides that are the amino acids that give flavor potential to your product. The process is so easy as you can see in the figure. You put the fishes or the byproducts in a reactor, you add the enzymes, you control the pH, and you can see how the enzymes act and produce from this fish a kind of hydrolysate that is a liquid that you can separate after that with centrifugation. That is your product. This is basically the flow chart of the process of hydrolysis to obtain peptides, but in the same process you have some co-products that you can see that are the bones of the fish that remain as a solid fraction that is not digested, that you can recover and use to produce mineral supplements, for example. You have some solids that are finally not digested, that can go to the fish meal production. You have fish oil that can be separated, and fish oil can be also valorized. And finally, you can obtain this peptide concentrate as flavor or as bioactive product. Using also the technology we have seen, that's the filtration to concentrate and finally evaporate and dry and obtain a product that can be a concentrate, a liquid, or can be a powder. And as we have seen in the previous presentation, we have worked with whole fish, hake or herring to obtain bioactive peptides, and we're working still to obtain flavors from these liquid side streams from the mussel production, and also using byproducts or solid byproducts to obtain bioactive peptides or flavors through this hydrolysis technology. And this is from my side. Thank you, and I leave the floor again to Mehdi, who will continue.
I will talk a bit faster, but Carlos explained a little bit about the hydrolysis technology, where we break down proteins into peptides. But here I will talk a little bit about another technology, which allows that we extract intact proteins, which will have different applications. I will skip this. You heard a lot. What is the challenge? But one thing is that, of course, how we can extract these proteins and valuable ingredients that are in this side stream that are generated. One example was the hydrolysis technique that you heard about. But I'm going to talk, as I mentioned, about another technology, which is called the pH process, where we grind the side streams, whatever that comes, all together. We mix them with water, and then we get a soup, like the first bottle that you see in this picture. Then we adjust their pH. The proteins will be solubilized. With a centrifugation, you can separate the solubilized protein and fat from non-soluble part, which would be the bone. You see in the lower picture what will happen. The protein will be separated from the bone and fat. Then you can readjust the pH of the stabilized protein, and you can dewater them. Then you will get a mince or a paste like this that can have different application. We have tested it in many products like burgers, fish balls, fish sausages, so on and so forth. It would be endless application like a surimi substitute or mince substitute. It can be also dried, and it can be used as an ingredient in emulsion-based product, in soups, in stews that we have tested it. Or it can be used as a liquid and injected back into the fish fillet, or as a coating, or in a battered product as a butter. What would this mean for the industry? It would, of course, result production of multiple products from the side streams. It can be applied in different types of side stream from different resources. We have also tested on many different types of resources. It will make profit from what is already lost from the seafood industry. Of course, the technology is done completely at cold. No drying is needed. Much lower carbon footprint compared to the previous technologies. You will get more protein out of each kilogram of fish that is harvested. And of course, it would be a more sustainable seafood industry for everyone. I will show the example for cod that we have worked on in the WaSeaBi project, for example. We have tested different types of byproducts coming from cod. And you can see how the protein will look like. It would be a paste with a very high functionality. But in case of viscera, it would be able to form gel, which is important for some product applications. Here you can see the gels that are made from these proteins. They can produce self-supporting gels like sausage, or it can be directly used in a burger products. Here you see the example for herring. We have tested different side streams and their combination. The proteins that are extracted, more like the protein from tuna. And of course, the properties would be very much dependent to the type of side stream, and their stability would be much less in case of head. But frame is a very promising case for this application. We have recently done very successful pilot trial for cod and salmon. And in October, we will do a big pilot trial for herring. By that, I would say thank you, and I will invite the next person.
Okay. Okay, good afternoon. My name is Jone. As a member of AZTI Group, I will give you an overview of the market applications for seafood processing side streams, from nutraceuticals to food and feed. As we mentioned before, the overall objective is to build an efficient and sustainable valorization system of seafood side streams into marketable products. We have worked within the consortium with a lot of side streams from mussel shells and cooking waters, salmon frames, undersized hake, cod and herring brines, and gut, viscera, head, and so on, to obtain these valuable end products. The products have been protein ingredients, bioactive peptides, mineral supplements, and savory compounds. In the case of protein ingredients, we have worked with fish protein concentrate. Normally, in the market, we could find them as powder product with a protein content between 65 and 80% and could contain some level of fats. The main technologies that we have applied in the consortium have been pH shift, membrane technology and flocculation of brines and cooking waters, and also the hydrolysis of different discards and undersized catches. We have obtained a lot of different results of protein yields, protein content, and depending on this protein content, also the functionality, as Mehdi told, and amino acid composition, we can have different market application. For example, we can use it as protein ingredient or supplement in the food industry, as a nutritional supplement, and also as animal feeding, when the protein content on the value is a bit lower. In the case of bioactive peptides, these are protein hydrolyzed. We have maintained them as protein powder product with a protein yield higher than 70%, a content higher than 90%, and it contains a lot of amino acid and easily digestible peptides. Depending on the enzymes combination, we can have different peptides in length and also in composition, what could lead to different bioactivities, such as antioxidant, antihypertensive, anti-cancer, antimicrobial, anti-inflammatory, bone protector, immunostimulant, anticoagulant, and so on. We have worked with 3 of them with good results in antioxidant, antihypertensive, and antimicrobial bioactivities. And regarding the market application of these products, could be as protein supplement in the food industry also, like a supplement as health promoting properties, for example, in sport nutrition, and also in animal feeding, maybe to higher value market as aquafeed and pet food due to its higher digestibility and bioactivity. Therefore, there could be a suitable substitute for antibiotics in livestock production, could help, for example, to avoid resistance to conventional antibiotics. However, their most applications are in nutraceutical market. For example, here we can see different antihypertensive capsules and pills, and this is due to the high strict laws in the food industry to put these health claims in a food product. Next product are mineral supplements that come from fish bone products. As Carlos told before, they come from, it's a co-product from the enzymatic hydrolysis. We have maintained them also as powder product with between 30–34% of protein, 20% of calcium, 9% of phosphorus, and some of them also rich in zinc and iron. Depending on this composition and the bioavailability of these compounds, they can have different market application in food, feed, and fertilizer. We can find different products in the market with different prices depending on these characteristics. And the last product has been the savory compounds. We have also maintained them as powder product and obtained by membrane technologies and also by enzyme hydrolysis. These products have a high concentration of free glutamic acid that is related with this umami flavor. And the market application, the main market application is as a savory ingredient in powder or sauce format for food products because they are related with this umami flavor. And another application also could be when you apply this membrane technology, you are going to do is to remove or to decrease this chemical oxygen demand of these wastewaters. So then you can have this wastewater below the sewage discharge limits, and also a concentrate to use it as a every ingredient. Which are the next steps in WaSeaBi project? In the next months, we will perform these pilot trials to validate these technologies and valuable end products. We will perform also with this data, this techno-economic feasibility study together with the life cycle assessment, and then we will see how to bring these products to the marketplace. Thank you for coming. I give the floor to Grethe again.
Time for the last part of the Kahoot. I know that the time is running, so maybe we should just go for that, asking you to answer the questionnaire in the postcard, and thank you all for coming and listening to our project. And please visit the WaSeaBi homepage. There will be newsletters and also information about where the results already have been published. So thank you very much for coming. And if you have questions, just come with them to us either now or later. Yeah.